Hello. Boy, you're in luck today. We're going to talk about animal nervous systems. This is one of my favorite topics. I've always been interested in the nervous system and the brain and senses and things like that. So today we'll be discussing that for our lecture. So the nervous system is, one of the couple of things that control the human body or the animal body. One system that controls the body would be the endocrine system. It's a system that we're not going to really cover in this class, but this is a system that makes and secretes hormones, and those hormones will be released from endocrine organs, and then they'll have their activity in some other part of the animal body. And endocrine system, some of the endocrine glands can secrete things and they can work very fast, and other times it takes days to weeks for the endocrine secretions to actually work, the hormones to actually work. Now the nervous system is a little bit different. The nervous system is made for fast transmission. And when I talk about fast, 330 miles per hour is how fast a transmission or an impulse can actually travel down a nerve cell. So if you've ever stepped on a piece of glass before or touched a hot stove, it doesn't take you long to get your hand or your foot off of the sharp or hot object. Now, when you look at animal nervous systems at the cellular level, they're very, very similar. We really worked out how human nervous systems worked, or how human neurons worked, by studying the neurons of other species, because at the cellular level, they're very, very similar to each other. But as you advance to more and more complex animals, the organization of the brain and the nerves is going to be somewhat different. There's also different adaptations, different sense organs that help the different creatures live in different environments. For example, bats have the ability to make high-frequency sounds, and then they have the ability to detect those sounds and find where prey are while they're flying. Dolphins can have almost like a sonar effect, making noises and then sensing those noises. And then, of course, rattlesnakes can see prey at night because they have special electromagnetic receptors that allow them to sense heat coming off their potential prey items. And we have our own adaptations as well. These are just showing you some of the different kinds of nervous systems that exist. So one of the, I guess, more primitive, in quotation marks, primitive, still pretty complex, is one of the simplest nervous systems would be the nerve net found in cnidarians. Okay, and that's basically decentralized nervous system. There's no brain, there's no spinal cord, but it's a collection of nerves that extend out and help to regulate the body of the organism. If you look at starfish have what we call a nerve ring. So you can see how that's arranged there. When you get up into the planarians, the platyhelminthes, we begin to see a ganglion or even a small brain with nerve cords that extend out down the lateral sides of the body. Here's a leech, which is a segmented worm. And so it has a brain, and then it has a nerve cord that extends down the center of its body. So we can see that the insect has a brain, and it has nerve cords, and it has ganglia, which are concentrations of nerve cells that extend throughout the rest of its body. So, when we get into the mollusks, some mollusks like octopuses have very, very complex brains, and then they have axons that come out, which are parts of their nerve cells that go out and extend throughout their body. And then when we get to chordates, animals that are vertebrates with a backbone, we get more and more complex brains, and we get a spinal cord, which is a collection of nerves that extend from the brain and connect the brain to the body. Okay, so over evolutionary time, we've seen that things have gotten more and more complex. When you get up to the vertebrates, mammals, and salamanders and lizards and things like that, they get really super complex. The starfish is a little bit of an anomaly. It's a very complex organism. It's a very advanced organism, but it's actually decentralized its nervous system to be more simplistic. So which is better, being simple or being complex? Well, each of those are around today. They've survived the test of evolution, and so they both in their own right have made it, and so who's to say which is better to have? Okay, so the nervous system is made of nervous tissue, as we talked about before, and the processors and sensory information gatherers and the and the nerve cells that conduct nerve impulses are called neurons. Neuroglial cells are going to be the other types of cells that support the neurons. So if we take a look, the large cell that you can see here is a neuron, whereas every one of these little circles you see here are the nuclei of all the neuroglial cells. And in like the human brain, you have a 10 neuroglial cells to 1 neuron ratio. So a lot of nervous tissue is made up of those neuroglial cells, and they have different kinds of jobs that they perform for the nervous tissue. Some of the neuroglial cells are going to, are going to make cerebral spinal fluid, like these ependymal cells up here, which line fluid-filled cavities called ventricles of your brain. We have microglial cells, which are like the little vacuum cleaners. They vacuum up cellular debris and microorganisms that may have entered into the brain. The astrocyte, which is this structure, this cell right here, basically it's going to support neurons, so it forms a scaffolding inside the brain and supports neurons, but it also forms what we call the blood-brain barrier. This blood-brain barrier is a barrier so that poisons and different things that are inside of your bloodstream, they can't get into the brain unless they go through these little processes of the astrocytes. So these little processes are very selective. Okay, and they help to protect you from ingesting certain kinds of poisons. Unfortunately, if you ever get a cancer in the brain or know someone with a cancer in the brain, it's the blood-brain barrier that prevents many chemotherapy chemicals from actually allowing people to take chemotherapy to treat a brain tumor. So, you have to have, for some brain tumors, you have to have it surgically removed, or you have to have radiation in order to get rid of the tumor, because chemotherapy won't come through the blood-brain barrier. Well, the oligodendrocyte is a type of neuroglial cell that wraps around the axons. Axons are little processes that come out of neurons that communicate a signal, and you can see that these oligodendrocytes are wrapping around the axons of this neuron, and this acts as insulation to speed up the impulse. If an axon is not myelinated with this fatty sheath around it, the impulse goes much slower, maybe 2 miles per hour. But if you have myelination, it actually goes something in the order of 330 miles per hour. So that's a pretty significant difference. There are all kinds of diseases where the myelin wears out, and that causes you to lose control of your muscles. And another thing is that when you learn and when you practice, probably some of you play musical instruments, or maybe you dance or do perhaps a martial art like karate. When you do those activities, you actually create new myelin and become better and better at doing those particular activities. So hopefully you are creating lots of myelin in this class because you're studying hard and doing all your work. Okay, so let's talk about neurons for a few minutes. Neurons are very special cells because they have the ability to change their voltage. They're excitatory, and I'm going to show you how that mechanism works. Now, there's 3 major types of neurons. There are sensory neurons. Sensory neurons are going to collect and send information to the brain and spinal cord about what's going on inside your body and what's going on outside your body. So your eye, your ear, your nose, your tongue, these have lots of sensory neurons that collect information and send it to the brain. But there's also sensory neurons in your skin and inside of your muscles and your joints, and those too communicate information about what's going on inside the body, and they send that information to the brain. There are inner neurons. Inner neurons are going to be basically neurons that you find in your spinal cord and brain, and they're going to process information and send messages to direct the next type of neuron called a motor neuron to tell the body what to do. So motor neurons are going to carry nerve impulses from the brain and spinal cord to the muscles so that you can voluntarily control them, and to different glands of your body. so that you can control those. And this is just showing you kind of how the relationship between interneurons, sensory neurons, and motor neurons, just showing you a simple reflex arc. So over here we have a sensory neuron that's located inside of your muscle tissue. So if a doctor or somebody hits you with a rubber mallet and it hits this little particular tendon, it'll stretch the muscle here, and there's a sensor that will sense that. That sensory information will go to the interneurons that are in the spinal cord. So this is a little piece of spinal cord here. And those interneurons will process the information, and they will command a motor neuron to do some kind of activity, like perhaps flex your muscle in this particular direction. And this is a simple reflex arc. You know, I don't know if you ever thought about it, but your spinal cord thinks too. It thinks involuntarily for you. So we have lots of different kinds of reflexes where you don't really need to think about those activities, but your spinal cord does, and it has interneurons that process that information and will dictate to a motor neuron to move a muscle or to do some activity. So that's kind of the relationship of sensory, inter, and motor neurons. So remember, your interneurons are in your brain and spinal cord, sensory neurons are all throughout your body, and motor neurons extend all throughout your body as well. Okay, so these things we call neurons, even in simple animals, are going to have very similar parts. They're going to have dendrites. Dendrites are these little processes. It almost looks like a tree. You know, the root dendra means tree. And so these little processes that you see all around this neuron cell body, these are called dendrites, and they receive information from other neurons. So information can come in through the dendrites and travel through until it hits the axon over here. So the cell body is just the main body of the neuron, and of course it has a nucleus. There is a conical area, and it's called the axonal hillock. And so any messages that come into this neuron are going to eventually reach the axonal hillock. And the axonal hillock will grade down into this little cord called an axon. And this cord extends all the way down, and it terminates here into these little synaptic knobs that we talked about when we did the muscular system. And these synaptic knobs, of course, have synaptic vesicles, which have neurotransmitters. Now, this particular neuron is myelinated. It actually has the myelin sheath, so it actually has insulation that surrounds the axon. But you could have an axon without myelin, and that would be an unmyelinated axon. And you can see over here there are two nerve cells that are basically contacting each other. They don't touch each other, but the synaptic knob comes really close to the next neuron, which has receptors that can receive information from the neurotransmitters that are dumped. So the electrical signal doesn't jump the synapse, this little space right here. It actually comes down and stimulates calcium channels to open up and allow synaptic vesicles to dump their products into this little space right here. All right, so what I want to do is talk to you a little bit about physiology of how the neuron actually works. So what I've done is I've shown you a picture here of the axon, just showing you a little piece of the axon here. And you might ask, well, you know, how does it send a signal from the dendrites all the way down to the synaptic knobs down here at the end? Well, that signal is basically a change in charge. So for the most part, there's a negative charge on the interior of the axon, and I'll show you how that's established in just a second. That voltage is about negative 70 millivolts. But because there are proteins sitting in the surface of this axon right here, we do have the ability to flood the inner part of the axon with positive charge. Okay, so you can see I can flood it with positive charge, and that's how a signal is sent down an axon. It's just a wave of positive charge that flows in. Now you have to have the ability to stop the flow of positive charge in, and I'll show you how that's done in just a few minutes. But you know, we can take and we can change the voltage of this from being negative to being positive, and then we can change it back to being negative again as well. So we can send a signal down an axon, and once it reaches the end of the axon, when it gets to the synaptic knob, neurotransmitters will be dumped out to continue the signaling to the next cell. All right, so I've drew a blank screen here because I wanted to show you how complicated that actually is. So you want to follow your study guide for how much of this you need to know. So I'm going to draw an enlarged axon here, and I want to show you the things that are in play to allow for this voltage change that occurs in the axon. Now, there are really a few parts that you need to know. So I'll just draw these particular parts right here. But to maintain negative 70 millivolts, you actually have to have proteins inside of the membrane that are going to help to actually control that. So we have this particular channel right here dumps potassium outside the cell. And so this is a potassium channel. And we call this a leak channel because it's always leaking potassium from the inside to the outside of the axon. And we have lots and lots and lots and lots of these. Well, you can see if you leak out a lot of positive charge, you are going to, basically become negative. Now, this is a sodium leak channel, and it works this way. So sodium comes in this way, and we have very few of these particular leak channels. Okay, so we have a lot of potassium that's leaking out, but very little sodium that's leaking back in. And then we have this thing called the sodium-potassium pump. And this is a pump that uses ATP, uses energy, and what it does is it pumps 3 sodium out for every 2 potassium that it brings in. And it works all the time. It uses ATP all the time, and it works all the time. Now you can see that if you lose 3 positive charges and only bring 2 positive charges in, that's a net loss of a positive charge. So if you take into account all the positive potassium that's leaking out, all the sodium that's leaking out, and the very little bit of sodium that's coming in, this helps to maintain that negative 70 millivolts. Now, every cell in your body has these particular things that are happening to it, and especially the sodium-potassium pump, and that maintains a negative charge right on the interior of this thing that we call the axon, the extension out of the cell membrane or the extension out of the cell body of the nerve cell. In addition, the neuron is very specialized in that it has a couple of extra proteins that are channels. So it's going to have a voltage-gated sodium channel, and it's going to have a voltage-gated potassium channel. Okay, so this is the way that the voltage-gated sodium channel works. This is the way the voltage-gated potassium channel works. Now, if I take and open up this voltage-gated sodium channel, I can flood the interior of the axon with positive charge. But if I open up the voltage-gated potassium channel, I can basically take positive charge out of the cell. Okay, so I can regulate if I want the cell to be negative or if I want the cell to be positive by altering these particular proteins here. Now, these are always working down here, but these I can stimulate to work. Okay, and therefore get a way to send a signal down the axon. Now, we can take an axon, like a giant squid axon, and we can stick little micro probes in there that can sense the electrical charge. Okay, so I can stimulate this with drugs, I can stimulate this with different kinds of poisons, and I can look at the charge change. I can see if it's a positive charge change or a negative charge change. And I can actually look at this on an oscilloscope and look at it on a computer screen. So an oscilloscope is mainly a computer screen that can graphically display what information we're seeing down here inside of the axon. Now, there is a y-axis and an x-axis. So the x-axis has time in milliseconds, and you can see it goes from 0 to 5. So 5 milliseconds, 5 thousandths of a second. And over here we have on the y-axis millivoltage. Okay, these are millivolts, thousandths of volts. So we have negative 70 and positive 40. A lot of times you'll see positive 30 on some of these graphical displays. So at rest, most of your cells are at negative 70 millivolts, and they don't change from that. But special cells like nerve cells and muscle cells, they have the ability, if they get a stimulus, they have the ability to open up those voltage-gated sodium channels and actually cause a positive charge to flow into the axon, and that's called depolarization. So notice we're going up in voltage, we're becoming more and more positive as sodium flows into the axon. So as sodium flows into the axon, the charge changes to being more positive. Now, at the same time that stimulus opens up the sodium channels, it also begins to open up, but very slowly, the potassium leak channels. So at the height of this depolarization, the sodium channels will eventually close. They open for a very short period of time and then they automatically close. And when they close, sodium stops flowing in. So we stop flowing the sodium in. But when we open up those voltage-gated potassium channels, then positive charge is going to flow out of the cell and we will repolarize. That is, we'll go back towards being negative because those potassium channels are leaking the positive charge out of the axon. And those potassium channels will open up for a period of time, even beyond once they get to negative 70 millivolts, and we get this thing called the refractory period. And then once the potassium channels close, then the leak potassium channels, the leak sodium channels, and the sodium-potassium pump will actually go back and maintain negative 70 millivolts until a new stimulus will come and stimulate an action potential to occur. So this is an action potential, and you can graphically see it. Because we have the ability to put it on an oscilloscope. Okay, so if that seemed confusing to you, you may want to just like rewind the video, listen to it again, rewind it back and listen to all the different little proteins on there. I'm also going to give you a series of notes that give you exactly step by step what occurs there, just to refresh your memory from what we've already stated there. Okay, and this is your steps to the production of an action potential. Remember, the action potential is the signal that is going to be sent down an axon so that we can communicate a signal from one nerve cell, and eventually it ends at the synaptic knobs, so we can dump neurotransmitters out and communicate to the next cell, or communicate to a muscle cell for that matter. All right, so let's look at these steps to the action potential. So at the resting state, and you can go back and look at your graph or look at it in your notes right there that you have at your desk. At the resting state, the sodium channels and potassium channels, that is the voltage-gated sodium channels and the voltage-gated potassium channels, are closed. Now all your leak channels are working and your sodium-potassium pump is working, and that's why you have negative 70 millivolts. Now, if you get a stimulus that is strong enough, it will begin to open up those voltage-gated sodium channels, and sodium will begin to flood into the interior of the axon. And that sodium will cause more sodium channels to open up, because as you get a stronger positive charge, it'll open up more voltage-gated sodium channels. So as you get more and more of these sodium channels opened up, you get a stronger and stronger stimulus, and eventually it will trigger a full-blown action potential to occur. And if you look at it graphically, that action potential, you'll begin to see it rise. That is, more positive charge will flow in, and it'll begin to flood the interior of the axon, and it makes the membrane positive compared to the outside of the axon. Now, this stimulus also opens up voltage-gated potassium channels, but the voltage-gated potassium channels are slow. So the sodium channels get a good millisecond or so to work before the potassium channels are opened up. So the positive charge floods in very, very rapidly. And we're not talking about opening one channel. We're talking to open thousands and thousands and thousands of channels. So lots of sodium has the potential of flowing in very quickly. Now, when those sodium channels eventually close, because they don't open very long, so as soon as the stimulus opens them, they only have about a millisecond, and then they close. So once those sodium channels close, you get the flow of positive charge stops. You get potassium channels now are catching up, and they're opening up, and they're allowing positive charge to leave the axon, and this is going to cause the inside to start to become more negative, and that's why we call that the falling of the action potential, because it's falling back towards being negative. And if the potassium channels remain open for a period of time, then you'll go below negative 70 millivolts. So if I just graphically look at this, this is the undershoot right here. So this is negative 70 millivolts right here. So if I keep those potassium channels opened up, more positive charge will flow out. And but eventually, that stops working. And once the potassium channels eventually close, it allows the resting state potential, or negative 70 millivolts, to be reestablished, because you have all those leak channels that are working and you have the sodium potassium pump that's working. Okay, that's probably pretty complicated if you've never had that before, but I believe if you go back and rewatch this and work with your notes a little bit, I believe that that it will all come together for you. But if you have questions, feel free to email me and we can go over that again. And this is showing you, just a little bit, it corresponds, each numerical value there corresponds with a graphic to show you what's happening with the sodium channels and with the potassium channels. So at resting state, they're both closed. If you meet the threshold, if the stimulus meets the threshold, then you can see graphically displayed over here, the sodium channels begin to open up and sodium flows into the cell. But the potassium channels are slow. So if you see the rising phase, the depolarization, we have all our sodium channels opened up, but the potassium channels are closed. But, sodium channels now are closing, and finally the potassium channel wakes up and opens up, and we see positive charge flowing out, but not positive charge coming in anymore. So we get a falling of the action potential because negative charge is now taking charge. And then if you keep those potassium channels opened up for a period of time, we'll get hyperpolarization. Eventually, all the channels will be closed and you'll go back to resting state, negative 70 millivolts, because of the leak channels and because of the sodium leak channels and because of the sodium potassium pump working. Now, we can graphically look at this, and if we stimulate with drugs or poisons, if we stimulate potassium channels, it will cause a hyperpolarization. So if you've ever had something like Novocaine before, like you had a cavity and you had an injection of Novocaine into your gums, what Novocaine does is it stimulates the opening of potassium channels, so you cannot create you stay in a state of hyperpolarization, so you cannot create an action potential. Therefore, your sensory neurons that sense pain can't send information to the brain because they can't generate an action potential. And that is a wonderful thing that we have medications that can do that, so we don't have to experience pain all the time. But there's other poisons and drugs that can actually stimulate sodium channels to open up. There are many poisons that stimulate sodium channels to remain open, and they cause your muscles to contract and not stop contracting, and that causes spasmodic paralysis. If I take a poison that won't allow sodium channels to open at all, then I can stop the muscles from working and cause what we call a flaccid paralysis, where you can't move your muscles. And that's a good thing too, that we have drugs that can stimulate these things, and also we can shut these things down. You don't want to twitch around while you're doing surgery on having surgery done on yourself, so we can take and paralyze your muscles with these things. And you've probably heard of Botox before. That's a muscle, it's a muscle it paralyzes muscles so that they don't cause wrinkles to occur. All right. So this is just a full-blown action potential here that we've talked about. So let me just talk about saltatory conduction real quickly, just to refresh your memory. If you have myelin, you have the ability to send a signal down the axon much more rapidly than you would if you had a non-myelinated axon. Non-myelinated axons have to flood the cell with sodium all the way down the axon. So every one of the little channels has to open up, and it's really slow to proceed through. If you have myelin, though, myelin gives you the ability to jump from space. So if this is myelin here, and this is myelin here, and this is myelin here, on only the channels that are here have to open up, and then only the channels here have to open up, and then only the channels in between these cells that are creating the myelin have to open up, and you get a very, very fast action potential that travels somewhere in the order of 330 miles per hour down that axon. Okay, so if you ever lose myelin, or you ever know somebody that has a disease where the myelin has been destroyed, multiple sclerosis is very common now, and that's an autoimmune disease that removes the myelin in nerve cells in your brain. And people that have that particular disorder, or other disorders where myelin is destroyed, they lose the functioning of various things in their body, especially their muscle tissue. And we've looked at this before, but I just want to refresh your memory. As the action potential travels down, and we looked at this in the muscle lecture, but you have sodium channels, voltage-gated sodium channels, that allow sodium to flow in. So as the action potential comes down, there's a voltage-gated calcium channel that will allow calcium to flow in, and that will be the signal for synaptic vesicles, which are holding neurotransmitters, to dump the neurotransmitters into the synaptic cleft. Those neurotransmitters will go down, and they'll contact channels down here that receive the information from that neurotransmitter, and they'll begin to flood the next cell with positive charge. Okay, and that's a simplistic view of it, and there's all different kinds of neurotransmitters that work in different kinds of ways, but we're just trying to get a sense of how this nerve cell can communicate to this nerve cell down here. All right, so you might want to go back and refresh your stuff we talked about when we did the muscle lecture. And this is just showing you a graphic of the little receptors that receive the neurotransmitter. If the neurotransmitter touches that receptor, it forms a channel and allows sodium to flow in. And we talked about last time in the last lecture that there is an enzyme that can enzymatically degrade that neurotransmitter, thus closing the channel so that sodium can no longer flow in. We have to have the enzymes so that we can stop a signal from continuing, okay? Because that could be very problematic. It's really cool, but this particular space right here is where many different kinds of poisons and venoms work. Sometimes venoms keep these channels closed indefinitely. Sometimes venoms open these up. If venoms close these off and they're going from motor neurons to muscles, then if no sodium can flow in, then you get flaccid paralysis. Basically, the prey suffocates that was envenomated with that venom. Sometimes prey will suffocate because they can't stop contracting. Sometimes prey, suffocate because they can't contract at all. Either way, snake eats, or whatever venomous animal injected that venom eats. In the nervous system, you could spend your life studying the nervous system. We don't know hardly anything about it, but it's not like a one-to-one ratio, like one nerve cell contacts one nerve cell, but you might have hundreds to maybe 10,000 nerve cells that communicate with one nerve cell and try to influence what it does. Okay, some nerve cells are excitatory, and they give a signal to tell this nerve cell to send a new signal down the axon. Some nerve cells are inhibitory, and what they try to do is inhibit this nerve cell from sending a signal down the axon. And so it's whoever wins. So if more excitatory nerve cells are communicating to the cell than inhibitory, then the signal will be sent. But if more inhibitory are sending signals to this nerve cell, then it will be inhibited. And it's good that we have inhibitory and excitatory nerve cells trying to exert influence. It allows us to turn on or turn off a nerve cell. Okay, so let's move on and talk about some of the growth structure, the anatomical large parts. So the animal nervous system, if you get into complex animal nervous systems, does have a central nervous system made up of a brain and spinal cord, and it has a peripheral nervous system composed of spinal and cranial nerves. So if you look at the brain and spinal cord, the brain and then the spinal cord, these parts make up what we call the central nervous system, and they have the interneurons, and they're the processors of the nervous system. But spinal nerves and cranial nerves are really important. There are little nerves that come out of your brain, and then there are nerves that come out of the spinal cord, and these are going to make up the peripheral nervous system. These are the things that are going to carry information to the body and carry information from the body to the spinal cord and the brain. So the brain is very complex, and this lecture is not going to get into the deep complexity of it, but your brain is composed of different parts anatomically and functionally. So the outer part of the brain is called the cerebrum. So what you're seeing in this particular picture right here, this big portion right here is the cerebrum. And in humans, it's really large, and other animals, it's not so big. We then have the diencephalon, which is going to be located kind of in this portion of the brain right here. We have the cerebellum, which is the back portion of the brain. Cerebellum means little brain. And then we have the brain stem that's located right here, which is made up of the midbrain, the pons, and the medulla oblongata. And then we have the spinal cord coming out of the back end over here. So each part of the brain has a different job that it performs, and we know that because you can take and you can saw a person's head or cranium off, and using local anesthesia that you inject into this area, and you can be talking to the patient after you cut their head off or cut their cranium off. You can talk to the patient, and then you can take little electrodes and stimulate with electricity different parts of the brain, and we can actually see or talk to a person and ask what they are sensing, or what they are hearing, or what they're tasting. So we've made a map of the brain to show us all the different functions of the brain, because of that weird quirk that the brain itself doesn't feel pain. Only the muscles and the tissue around the brain feel pain. So you can use local anesthesia to numb that up, and then talk to a person while they're still awake. Okay, so what I want to do is talk about a few of these parts with you, just to give you a sense of what's going on here. So here's a simple map that was made of the brain to show you some of the different regions of the brain and the different functions that they serve. So the cerebrum or cerebral cortex is made of different lobes. We have the frontal lobe in the front. We have the parietal lobe back here on the side. We have in the very back the occipital lobe, and then we have on the side the temporal lobe. There's a lobe called the insula that's inside, but you can't see it in this particular graphic. So some of the things that the frontal lobe does would be things like in this area here, speech. In this area here, this is what coordinates all of our muscles. We also have over here ethics and higher, mathematics, abstract reasoning. These are all in your prefrontal cortex, good decision making, problem solving. Those would be where stuff would occur. We sense all of the things that we feel in the somatosensory cortex. This is a region here where we taste. All of our hearing comes from areas of the temporal lobe, and smell also comes from the temporal lobe as well. We see from the occipital lobe. How do we know that? We stimulate with electrodes different parts of the brain, and we've made a map of that. So this is the motor cortex over here, and this is the somatosensory cortex over here. The motor cortex is the cortex of the brain that allows us to move. And using those little electrodes and talking to people while they're awake during brain surgery, we've mapped out that your tongue muscles are actually controlled by that part of the motor cortex. So it's located right about there. And all of your facial muscles are located right about there. And your hand is located right about there. And then the rest of your body is mapped on here. So you have a left side and a right side of your brain that controls the right and left sides of your body. But just as we have a map of the motor cortex, we have a map of the somatosensory cortex. So you actually sense your face here, and you sense your lips and tongue here, and then your hand is located right about there. And someone made a map of this, or made a little clay model of this called the somatosensory man. And what they did is they took and they mapped, they made a human model based on the proportion of the brain that was dedicated to that particular body part. So if a body part is highly or largely represented in the brain, we made that body part bigger, and if it had very little representation, we made that body part smaller. So by looking at somatosensory man, you can see that that a large part of the brain is dedicated to our lips and our tongue. That's why it hurts so bad when you burn it with your hot Starbucks coffee or latte or whatever you drink. Our hands are very sensitive because a large part of our brain is dedicated to sensing that, and our feet are very sensitive as well. But our back and our arms are not as sensitive, okay, and the back of our head's not as sensitive. Let's see if I can put my face over here. Well, another region of the brain is called the diencephalon, and it's made of all these parts, but there's only two parts I really, care for you to really understand at this level, and that's the thalamus. The thalamus is a relay station for all sensory impulses coming into the brain, and the thalamus is a really important part. You actually have two of them, and this is one of them, and this is the second one over here. And so all information that comes in from your senses of your body, except for smell, goes through the thalamus, and the thalamus chooses to send it to higher brain regions and make it into conscious thought. Another region of the brain that's really super critical is the hypothalamus. The hypothalamus is really critical because it has all kinds of parts that help to maintain homeostasis. So it maintains hunger and thirst. It maintains the water balance inside of your bloodstream. So it has many different influences, like creating body temperature or maintenance of body temperature for you. I didn't have a good picture here that shows the thalamus, but if I slide on back, I'll just show you that the thalamus is located right here inside the brain. So it's deep inside the brain. I meant hypothalamus on that, my apologies. The thalamus will be located right here. So these are pretty deep inside the brain. The cerebellum, or little small brain, located on the back surface of your brain, is involved in different kinds of parts. This is just kind of generic. It's involved in learning, decision making, consciousness, self-awareness, sensory awareness, and if you ever catch a baseball, it coordinates reflexes, sensory information coming from your eyes and from the proprioceptors, those receptors inside your muscle tissues and joints that show or tell your brain where in 3 dimensions your body is. It coordinates all that information and then coordinates your skeletal muscles to be able to catch an object. Catching an object is a very complex task. Well, really anything's complex that you do, but that's a really super complex task that you do. And the cerebellum's involved in that. It's involved in a lot of different things. I'm sorry to be so generic with it, but there's only so much you can learn in an introductory class like this. The brainstem is going to be made up of the brainstem, the midbrain, the pons, and the medulla. The midbrain is, you can see in the graphic down here, is located right here. And it coordinates some of your visual and auditory reflexes. If I were to stand behind you and yell and you get that startled reaction, that is coordinated by the midbrain. The pons serves as a relay station for information coming from the spinal cord to the brain and from the brain to the spinal cord. It also regulates the rate and depth of your breathing. That is not a place that you want to have a tumor because you stop breathing. The medulla is going to be the lower portion of the brainstem, and it regulates heart rate, regulates breathing, blood pressure, swallowing, vomiting, digestion. It is a really super important part at regulating all kinds of involuntary activities. Now your brain doesn't slosh around because there's meninges. These are coverings that surround the brain. They nourish the brain and anchor it to the cranium, so it doesn't slosh around. Now it's not perfect. You can get a concussion, by slamming your head too hard against something. But for the most part, when I just do my head like this, the meninges hold it in place and allow it to survive those particular movements. Your brain is floating, so it floats on a fluid called cerebral spinal fluid. You can see the fluid right here, And so you can see this fluid surrounds your whole brain. Your brain is floating. And but the meninges help to anchor it to the cranium so that it doesn't hurt itself. But again, if you don't wear your seatbelt or you get in a motorcycle wreck or certain sports injuries, collide with people, football and stuff like that, when you collide with people, it can overwhelm the ability of the meninges and the water cushioning to do its job. So let's talk about that peripheral nervous system to kind of wrap things up. The peripheral nervous system is, again, made up of your cranial nerves and your spinal nerves. And these are divided into several different systems. The somatic nervous system is a voluntary system that allows you to control your skeletal muscles. So it's the cranial and spinal nerves that help you to control your skeletal muscles. And that's under conscious control. There are also some skin glands that are in the somatic nervous system that are not in your voluntary control. So there's glands in your skin, sweat glands and things, where when you lie and you sweat, the somatic nervous system will be controlling that. The autonomic nervous system is going to be an involuntary system. It's not to say that you can't learn to control it, because there are people that are yogis or super meditators that have tapped into controlling their autonomic nervous system. But for the rest of us that don't do that, it's involuntary. And the autonomic nervous system is your cranial and spinal nerves that are connecting the central nervous system to your body parts. The viscera would include heart, digestive organs, blood vessels, smooth muscles all throughout your body like the uterus or surrounding your blood vessels, and various glands of the body. That's just what your cranial nerves look like. If you ever take an anatomy class or you go into the medical profession, you will definitely need to know these particular nerves. Probably most of you have had a cranial nerve test, but you didn't know what was happening. So if you ever go to the eye doctor and they ask you to follow the finger, so I don't know if you've ever followed the finger before, that's actually testing some of your cranial nerves. So, and when they press on your shoulders and ask you to clench your jaw, it's again testing some of these nerves. Each one of these nerves has a different job. So olfactory does, smell, and optic does vision. Oculomotor and trochlear, are going to control eye muscles. So facial controls your feeling of your face, and glossopharyngeal controls your tongue and your pharynx or your throat. Vagus is controlling your digestive organs, and hypoglossal controls your tongue. Vestibular cochlear is going to control your hearing and equilibrium. So those are cool to learn. If you ever take an anatomy class, you'll enjoy learning those. And these are just your spinal nerves coming off the spinal cord. I was just going to show you what they look like. So the autonomic nervous system is further subdivided into the sympathetic and the parasympathetic. The sympathetic nervous system generally prepares you for stressful emergency situations. So we're talking, Rottweilers chasing you and they're hungry and they're out for blood. That's sympathetic nervous system that will prepare you either to run or to just, go fetal on the ground. Parasympathetic nervous system is generally activated when you're just normal and you're resting. So that's when those are actually activated. If you meditate or do deep breathing or yoga, you are trying to activate the parasympathetic nervous system to allow your body to chill out. So when you are stressed out, you're activating the sympathetic nervous system. And they have different jobs that they perform. For example, the parasympathetic nervous system will stimulate your salivary glands. So when you get hungry and you get ready to eat, the parasympathetic nervous system is helping you to eat. It slows your heart. It constricts your bronchial tubes. It stimulates your stomach to want to digest food. It stimulates your pancreas to help you digest food. Okay, so those are some of the things that it helps you to do. The sympathetic nervous system, though, is preparing you to fight or to run away. And so what it's going to do is inhibit salivation. It's going to increase the size of the bronchial tube so you can get more air to your body. It's going to accelerate your heart rate, inhibit digestion. There's no reason to digest food if you're about to die. So that's some of the jobs of the sympathetic nervous system. And these two systems work antagonistically. So when the sympathetic nervous system is working, the parasympathetic system is not working. So, for example, if the sympathetic nervous system, it speeds your heart up, decreases secretions of the salivary gland, causes dilation of the pupil, whereas parasympathetic nervous system would have would have the opposite effect of that. Okay, well that finishes up the nervous system, the animal nervous system. So I hope you enjoyed that. I hope you learned something new. If some of the parts seem confusing to you, just rewind them. You know, work with your notes, look at it, do your reading. Make sure you keep up with your study guides. And if you have questions, please feel free to get up with me. Until next time, I wish you a good evening, a good morning, whenever you're watching this, and I'll see you next time.